What if the story of us wasn't carved in bone, but hidden in dust? In every cave floor and riverbank, DNA, albeit much degraded, clings to grains of earth. Could we learn about ourselves from those faint echoes? The surprising answer is yes. We call it sedimentary ancient DNA. It lets us see people and other life who left no skeletons, only genetic traces. Ancient DNA is transforming our understanding of human evolution and history. Yet core questions remain. What are we in terms of lineages, adaptations, and variation? Which parts of modern life are recent, and which are deeply inherited within our DNA? How large were human groups? How were they structured? And how did that change across geographic regions and times? When did cooperation, violence, and morality take their modern forms, and why? When did fully modern language emerge, and who did small groups speak to? The next leap comes from sedimentary ancient DNA, genetic traces that are preserved in caves, lakebeds, and soils. Even without bones, DNA from humans and other species binds to minerals and persists, though drastically degraded. Uncovering these layer by layer, we can identify who was present, when, and in what combinations. We can detect hominin lineages even in layers that lack skeletal remains. That is crucial for locating who lived in certain parts of the world in the past, for how long, and when and where intermixing occurred. Population transformation. It has already been established that populations can undergo major lineage change within a few generations, often not obvious in material culture. Only DNA reveals the complete transformation. Sediment ancient DNA can track arrivals, disappearances, and replacements as DNA signals change. Contact and overlap. We can identify co-occurrence and windows of interaction. Ecology and context. We can recover animals, plants, microbes, and pathogens to link people with climate and diet. Find time slices. Stratigraphy plus radiometric dating can tie DNA to specific dates. (450,000 to 40,000 years ago). Different kinds of humans lived across Africa and Eurasia. In Europe and Western Asia, Neanderthals adapted to cold Ice Age worlds. They made tools, hunted big animals, and lived in close groups. Farther east, the Denisovans survived in mountains and forests from Siberia to Southeast Asia. We know them mostly from DNA, not bones, but their genes still live on in people today. In Africa, Homo sapiens appeared and began to flourish. They made new kinds of tools, used symbols, and built wider social networks. These lineages developed in partial isolation, but occasionally interbred. These encounters likely influenced physiques and social behaviors, leaving long-lasting traces in our DNA. 70,000 to 10,000 years ago, humans spread into Eurasia, Sahel, and the Americas. Each expansion brought new challenges: different climates, landscapes, animals to hunt, and plants to gather. Populations adapted, languages and traditions diversified, and humans began shaping ecosystems across the planet. Last 10,000 years, agriculture and storage of surplus food changed the game. People grew food, stored it, and shared it. Surplus made some lives more stable, but it also brought new risks. Settled life tied people to land and harvests that could fail. And dense living brought new costs, crowd diseases, infections from animals, and shifting microbiomes. With agriculture, something new began: specialization. Not everyone had to find food. Some made tools, built homes, traded goods, or led communities. With time to spare, humans planned, built, and imagined. Villages became towns. Networks became states. Technology, art, philosophy, new human capacities emerged. Shared rules and trust held growing communities together. Cooperation scaled up. Diets changed too. More grains and starches. In some regions, milk and dairy. Over generations, our bodies adapted. New metabolism, new immunity, new ways to live. Advantages of synergistic collaborations between ctDNA and established fields. To move from method to meaning, we are launching partnerships with archaeologists who collect sediment samples in caves, open-air sites, and lake beds across diverse climates and time periods. As more sites and contexts are sampled, we gain statistical power. What seemed like fragments became a signal. The dirt turns from background to evidence, and the quiet layers of Earth begin to answer questions about how humans lived and evolved, even in the absence of bones. Why this changes our perspective. Sedimentary ancient DNA extends our record beyond rare skeletons to continuous environmental archives. It resolves migrations and bottlenecks with higher spatiotemporal precision. It tests social and subsistence hypotheses by coupling human signals with local ecosystems. It bridges archaeology, genetics, and paleoclimate into one timeline. From the Levant to the steppe, new timelines emerge. The ground itself becomes our archive, and it is rewriting human evolution.